this drug, we initially tested it in a rare but highly aggressive hematologic malignancy known as blastic plasmacytoid dendritic cell neoplasm, or BPDCN, which is known to highly overexpress the target of CD123. That drug was actually recently FDA approved for the indication that we studied for BPDCN. And based on that early success and encouraging results, we've now moved into investigation and testing of this agent in a number of different blood cancers. So based on this discussion, this led us to starting a phase 1/2 trial of this agent, tagraxofusp, in patients with myelofibrosis. The keys to this trial are that patients will have had at least one prior therapy, the majority of whom will have had a JAK inhibitor. Second aspect is that we've enrolled 23 patients so far that we're presenting here at ASCO. And then finally, the median age is 65 years, which is important as this does represent a so-called real-world or expected age group for patients that you would see outside of a clinical trial. So among these patients, another important aspect is that the majority of them have clinical features such as splenomegaly or symptom burden that we can then follow objectively over time. The safety profile for tagraxofusp is important to discuss. The main reason for that is we did note in the BPDCN experience the incidence of a potentially life-threatening condition known as capillary leak syndrome, or CLS. Now, among the 23 patients so far treated on the myelofibrosis clinical trial, we have seen capillary leak syndrome, but only in one patient at the high grade level, which is so-called grade level 4. So it is something to watch out for. It is something we understand, and one of the most important elements that can predict for it is the albumin level. So what you'll notice on all of these clinical trials with this agent is there's a baseline albumin level, and then monitoring the albumin and replacement of that as we move forward. For patients with myelofibrosis, there's a couple of important clinical compartments that you have to follow. One is spleen reduction, 2 is symptom burden reduction, and 3 is what's happening with the blood counts. This is an ongoing early trial interim analysis, but what we are seeing among these 23 patients is some encouraging results that need to be followed up with longer-term follow-up. So among these patients, approximately just under half the patients, 43%, with measurable spleens are seeing some form of spleen reduction. So that's encouraging. We need to continue to follow that and see how it goes over time. There are a number of patients who've experienced MPN symptom burden. Now, importantly, symptom burden is an objective measurement. It's called the myeloproliferative neoplasm TSS, or total symptom score, and that's a prospective assessment. Baseline survey of the patient, 10 most common symptoms, and then follow that over time. And on the poster, you'll see more of the details of that data set. And then finally, in terms of the cytopenias or blood counts, we know that with this agent, tagraxofusp, there's a transient thrombocytopenia that usually occurs in the first cycle. And again, that's what we saw here. This is not a major dose-limiting toxicity or problem with the agent. So as with any novel agent, in a phase 1/2 setting that's ongoing, there's a number of important future directions. For tagraxofusp in particular, I would like to highlight 2. The first is in the single agent experience, there is an early signal, but needs to be followed up with much longer follow-up, that potentially a poor subset of patients' prognosis is with monocytosis. So that's myelofibrosis with monocytosis. So that's different from CMML. There are emerging data that show that maybe those patients have a poorer prognosis than others, and with this drug being a CD123 targeting agent, the hypothesis would be that perhaps there may be a signal to target this, and we need to see that further. So in this clinical trial, as we're reporting in the abstract, the patients with monocytosis and splenomegaly appear to be having some clinical benefit, and so we need to follow that up over time. The second aspect is, what about combinations of therapy? That's what everyone always asks us in the field, and that's a good thought. Again, myelofibrosis is generally thought to be an incurable disease with chemotherapy alone, and stem cell transplant appears to be the only curative option. So in that setting, combination therapy is an attractive clinical approach for our patients, allowing the fact that toxicity profile is manageable and that there's some rational mechanism of action. So that's an exciting area of investigation, both in the lab and hopefully in the clinic one day, to be able to combine this agent with other rational agents in investigation for myelofibrosis.